My name is Bo Wang. I'm an assistant professor of bioengineering at Stanford University. I received the Beckman Young Investigator Award in 2017. It has been an honor and a privilege to be a member of the Beckman family. My lab hosts students with backgrounds in engineering, chemistry, physics, biology, and mathematics. Enabled by the generous support from the Beckman Foundation, we study a variety of animals to understand stem cells, regeneration, and cell type evolution. We believe strongly and deeply the scientific approach that Dr. Arnold Beckman took throughout his life, which is to emphasize on developing new technologies and tools for quantitative measurements in order to break down the intrinsic complexity of biology. A major focus of our work is one of the most prevalent human parasites called Schistosoma. These are small blood-feeding flatworms that live in our bloodstream as a pair of mating female and male. Each pair can lay thousands of eggs per day and can keep doing it for decades. Many eggs are trapped in host liver and cause inflammatory response eventually leads to death of patients. Remarkably, schistosoma eggs have been also found in mummies buried along the Nile River, and indeed many historians believe that one of the major reasons for ancient Egypt to fall were these little worms that disabled the whole society and dragged the people into poverty. A fundamental challenge to inhibit these parasites is that they have complex life cycle to multiply through two hosts, a molluscan intermediate host and a human definitive host. So hitting one point of this cycle is only partially effective, as the parasite can still survive at another life cycle stages. It has become widely accepted that a major target for disease eradication should be parasite transmission, which is driven by its life cycle. On the other hand, this life cycle is fascinating from a basic science point of view, as the parasite needs to deploy several drastically different body plans are specialized for infecting and producing each host. So we don't know how this parasites manage to do so. Over the past few years, through the support from the Beckman Foundation, we have used new technologies to sequence the mRNAs from individual cells isolated from the parasites and developed new analysis method that has allowed us to dissect the schistosome stem cell populations. As reported in a series of papers, we have identified stem cells that can directly reproduce embryos of new individuals to drive the parasite multiplication in the snail host. We found stem cells that are transmitted from the snail to the human host for initiating the next life cycle stages. There are stem cells that generate all sorts of somatic structures to support the vital functions of parasites so that they can survive in the hosts. We have also succeeded in capturing stem cells that give rise to the germline, which develops into sperms and eggs to restart the life cycle. Using functional genomic analysis, we have also identified gene regulatory programs that control the fate and the function of the stem cells. For example, the germline stem cells, the GSCs, need to be regulated to perform their two essential functions: proliferation, which produces small daughter stem cell through mitotic division, and differentiation, which initiates meiosis towards the production of gametes. We found that the maintenance of GSC's molecular identity requires an RNA binding protein conserved across the animal kingdom called nanos. Their proliferation differentiation is balanced by previously uncharacterized genetic circuit formed by a homeobox transcription factor 1 cut and an mRNA binding protein bool. Their expression are mutually dependent, and they inhibit collectively the overproliferation tendency of GSCs and promote differentiation, balancing the effect of another gene we discovered called ELID, which suppresses the differentiation of GSCs. Our work not only provides the knowledge base to understand and gain control over the schistosome life cycle, but also establishes the experimental and computational methods that allow us to go beyond one organism and examine stem cells in other related organisms. There are thousands of other flatworms related to schistosomes, and some were nicely documented by Ernst Haeckel in his famous book, Art Forms in Nature, about a century ago. Many of them are harmful, sometimes deadly parasites that infect human beings and other larger animals, and also propagate through various complex life cycles. We now can identify stem cells in many of these parasites and then compare their gene expression to understand how the molecular programs are conserved and diverged in stem cells from different organisms and how they evolve to address the distinct functional needs of these parasites. For centuries since Darwin, evolutionary biologists have relied on joint homologies in anatomy to compare organisms, and since the discovery of DNAs in 1950s, we have relied on gene homology to infer evolution. But we're now at an exciting turning point. With the new technologies, we can systematically compare cells and understand how they contribute to evolution, as after all, they are the basic unit of life.